Good morning, one and all. In the eighth session, in the first part, we'll be learning about the metabolic network. Till now, we have been extending our discussion onto like protein-protein interaction networks, and including the prediction of the answers, different kind of servers which are normally used. But now we'll be moving on to different other molecules which are involved in the biomolecular functional complete network in a cell. Okay. See, now extending our discussion further, there are three categories of metabolic networks. The first one is the metabolic pathway maps, okay, which just includes the flow of metabolites and different enzymes involved. Okay, just like a biochemical pathway. Okay, it includes all the CAC databases, different enzymes, different parts, all are put together. Just like the ecopsych, biopsych databases. Okay, all of them come here. Second one is the genome scale metabolic models. Okay, as the name suggested, genome scale. Okay, so for a gene across the complete throughout the genome, all the parts put together. Okay, so the genome scale, all these genome scale metabolic models (GEMs) integrate biochemical, genetic, and genomic data, so that what is the impact of different molecules on the overall cell can be predicted. Okay, it is used for metabolic engineering, drug discovery, and different other purposes. Third one is the flux balance analysis. Okay, the flux balance analysis. This part is analyzing the like how much the metabolic capabilities is there based on the metabolic network. So how much the amount you are taking up, the mass balance. We are normally doing the mass balance, that if this much mass of the metabolites is taken up for some certain set of biochemical pathway, how much output is coming up for different metabolites. Okay, we are doing the complete analysis. If certain metabolites are taken up per unit time, like certain amount of molecules should be produced also. Okay, it is used for also for several different metabolic models and allowing us to find the rate limiting enzymes. Okay, where the reaction rate drastically decreases. Okay, it has got several different purposes. Right. Now see here, in this part, like let me show you the complete part. All the different molecules, different enzymes are connected here along with the directionality. Like which enzyme is connected to what other enzyme. Okay. Now if you're zooming here from this to this, you're getting several different parts like biosynthesis of secondary metabolites, metabolism of cofactors and vitamins. Okay. All the parts are connected here. So what is the major emphasis? We can analyze the set of metabolic and like physical processes, how the things are worked out. Okay. This helps us to analyze, build a reaction model so that it can be used for the flux balance analysis also. All together in the network we cannot do FBA, but if we are able to minimize the data set like I have done it here, okay, this part I can now do it properly by considering pathway by pathway by pathway, okay, and for all this the tech, the string databases like these are helping me drastically. Okay, they help me substantially. Okay, all right. Now, to explain it from the nutshell, understand that the metabolism is defined as the totality of the chemical reactions that operate in a living organism or the cellular body, cellular system. All the reactions considered together, be it catabolism, be it anabolism, that is the be it breaking down of complex molecules to give you energy, or be it synthesis of newer molecules to get stored. Okay, just like if I'm taking some complex molecule, say like mag or any complex element, what happens the cell, the my body like stomach like uses the enzymes and the HCl to digest the molecules, like it breaks down the complex molecules into simpler ones to release energy. This is termed as metabolism. However, the anabolism is also there. Like once the my stomach is able to now like store the energy, and what form it is stored as, it will be using the anabolic reactions to like now join the small molecules into the bigger one, like fat. Smaller molecules are now integrated into the fat molecules and then they get stored. Okay, like all these parts, including NADH, ADP, here, ATP, NADH, NADPH, FADH2, all of them are within the entire all the cycles. That's a constant exchange or like used utility of energy or like synthesis of energy takes place. It has always been there in the cell. And all this builds up the complete set of metabolic reactions in a system. Now see, within all these different cycles, what exactly happens? The enzyme one interacts with the substrate one. This is just a typical diagram to show you how the things work. Okay, enzyme one interacts with the substrate one here. See, like then this part is fed to enzyme 2. Like this product is acting as a substrate to enzyme 2 here. Okay, then it pass on further. Like a substrate enzyme 2 product is fed to enzyme 3 as the substrate here. Okay, lastly the product is formed. This complete biochemical pathway on whole it's termed as if I'm analyzing the how the complete metabolic network works here, from the initial substrate, how much the mass is coming up per unit time, to how much mass is being produced here per unit time. Like all this will give me the flux balance analysis, the FBA system. All right. Now see, for example, I'm showing the glycolysis pathway here. Okay, I've taken it from the my molecular cell biology sixth edition book, the Freeman. Okay, this glucose is coming as a metabolized by hexokinase, giving rise to glucose 6-phosphate, the fructose 6-phosphate, and lastly it gives me pyruvate. Means glucose has C6H12O6, 6 carbon, is now broken down into two molecules of 3 carbon entity that is pyruvate. 3 into 2 is again 6. Okay. Whatever the mass I'm showing here, exactly the things are being converted here. See? 1, 2, 3. Exactly. Okay, the complete thing is here. In between, whatever the molecules are constantly being bifurcated here. Okay, the mass balance is constantly being going on along with the charge balance. Okay, so all these things give rise to FBA. And exactly when I'm putting up more enzymes, which are actually catalyzing all these reactions, it becomes a metabolic network as such. All right. If I ask why to study metabolism, okay, understand that in many patients, several diseases stand up. Okay, because of non-functional some enzymes. Okay, like sickle cell anemia. Okay, because in which RBC becomes sickle shape and their oxygen carrying capacity reduces. It is just caused by a small mutation. Okay, but it leads to an altered phenotype, altered mechanism, and altered reaction mechanism altogether. Okay, so if I am able to measure complete how much like uh, how much hemoglobin is able to bind the oxygen, I'll find that these kind of sickle cell anemia cell sickle cell RBC are unable to bind the required amount of this oxygen. There's some limitation there. Somewhere some problem is there. So by just analyzing the FBA here for these kind of cells, I'll immediately get to know some details in the mechanism. I'll get some insights. Like what is the estimate phenotype of a modified gene? Okay, so I can get some more information. I can dig out further. Okay, the metabolic enzymes and their regulators gradually becoming viable drug targets. So this is all going on for some disease cells, for some bacteria, virus, and these kind of things. I can devise a drug through which I can find the required cure as such. Why to stop? How to stop this reaction part? How to stop their problematic behavior? Likewise, if I'm able to know the metabolism, the another perspective is if I'm able to know how the enzyme is able to work, if I'm able to increase the activity so that the yield of the enzyme activity is improved, the enzyme is able to form much more product compared to the initial native enzyme. Earlier enzyme was only able to produce 2 mg per liter of the product, but now it has gone increased to 12 mg per liter, which is beneficial for me and the industry. So for all this efficient production of the biological products, okay, the all these metabolism parts, metabolic complete networking is very essential for me. I should be able to produce the build the reaction mechanism. I should be able to integrate all the enzymes, all the metabolites coming up in the picture, then build able to build a computational model how to work it out so that I can get the required details of the reaction mechanism. Okay. Now see, to understand it, to extend the discussion further, understand that the metabolite actually means the any kind of interacting atom, interacting molecule. Okay, like glucose, different oxygen, NADH, any part, any type. And the biochemical reaction is the one which is involving multiple different of metabolites through enzymes, and then it forms the required products from the reactants. Like in the last slide, I have been showing you the glycolysis pathway. But now see the same pathway with the perspective of ATPs and NADH, or how the cofactor of the enzyme is involved, why that energy is involved here. See the glucose is forming the glucose molecule is forming the pyruvate. The same pathway, same thing. But now, unlike the enzyme shown there, now I'm showing the how the NADH is involved or how the ATP is involved. Okay, how those things are involved. Okay, so see the complete part: glucose plus ATP, glucokinase is glucose 6 phosphate plus ATP. Okay, and all of them are like catalyzed through different enzymes. That is the entire beauty of metabolic pathway. All right. It's all lies from the perspective of different parts. Same reaction, same things can be analyzed through different strategies. Okay, but like we are all working out for the metabolic network, either through FBA, either through like computational models or different other things. Okay. Now to understand what we have learned so far, let us answer this small MCQ questions. Which of the following best describes the function of a metabolic network? Storage of genetic information, no. The control of immune responses, of course not. Regulation of regulation of cell division, this is also no. Interconversion of molecules and energy flow. Yes, we have been discussing all of this. Okay, that this is the right answer. Okay, now answer the second question. Which of the following computational models is commonly used to analyze and predict the metabolic capabilities of an organism based on its metabolic network? Okay. Is it DNA sequencing? Of course not. PCR? No. Flux balances? Yes. This is the right answer. Vaccine blotting? Not. While other three are the like basic molecular biology techniques. The third one allows us to analyze and predict the metabolic capabilities of an organism based on its metabolic network. So this confirms that we are understanding properly. Okay. Now answer it further. All right. Now answer this conceptual question. Is only catabolic reaction involved in the metabolism? Of course not, because we know that anabolism is also the required part. All right, we also need anabolism to work out different metabolic steps. Okay, so As I've given you the example of digestion, along with catabolism, we need anabolism to work out the complete cycle. Somewhere molecules are broken to form some other one. If that's then only the mass transfer or the charge transfer will take place properly. Some molecule has to break up. Then only some molecule can be produced. Alright. Alright, so now let us understand extend our discussion to the gene regulation. Alright. So because all these metabolic networks are not just connected as such. They are regulating many different networks, many different enzymes, so that the productivity of different products, different key molecules can be well regulated. So this regulation is governed through the metabolic network very precisely. Okay, only the required amount of product is formed under the given circumstances, and the system does not waste even a single molecule of energy. Now see, the gene regulatory network is very crucial for an entire cell because it is precisely regulating the metabolic control of all the different enzymes in the cell. Okay, regulation occurs at many stages, like pre-transcriptional, transcriptional, RNA editing, like how the different splicing takes place, or the translation initiation, post-translational modifications, RNA and protein degradation. At all these different steps, the complete control of all the different biomolecules is constantly taking place, so that different biomolecules, the concentration can be well regulated in a system. Okay, but understanding the regulatory process is the central problem of biological research. How the system controls it, or how under so many different enzymes, how the system is able to precisely regulate the proper biomolecular production of different enzymes under different circumstances. And even if something is produced, how it is degraded after some time. You understand, just before a cell cycle, many proteins of the cell cycle, okay, required in the G1, S, G2M, all these phases. These proteins are different, required molecules for this are always produced. Okay, they are just being produced at the required phase, just when it is needed. And after some time, the system again degrade it. Remember, metabolism, catabolism. The cycle keep on continuing monitoring. All the molecules are precisely produced at the right time, only when it is needed. Now, after that, the molecules are degraded. The system again restores the energy. Okay, and then these molecules produced are again utilized as the ingredients somewhere else for guiding some other pathway, and it keep on taking place. So now understand the beauty of all this nature. Without eyes or without even able to proper part, it is able to feel, sense. All the molecules are able to sense each other's activity very precisely and regulate the control. This control is very beautiful through which the entire system works. All right. See, in this slide, I'm showing you genes typically interact with more than one partner. Okay, multiple different proteins are constantly under control of several different enzymes, which are the key hubs. Okay, so major regulatory control is done by several different enzymes. Okay, so see, sometimes single input, single output. One protein is passing on the message to some other protein. Okay, and it's properly governed. But some other time, see, even if multiple inputs are there, it is leading to a single output. Only a single output is there. Like you might have heard of the example of the skin color. Skin color is properly controlled by three pair of genes, like which is termed as a quantitative inheritance. Like the dominant or recessive like allelic nature of each of these gene define the entire skin color quantitatively. Okay, like more melanin production is there or not there. Okay, but some other times, see, single input can cause multiple outputs. Okay, sometimes pleiotropic regulation. Okay, all of us might have studied in genetics, but now I'm showing you here for the so that you can understand the beauty of all the regulation, how it is properly governed, and all these parts comes under metabolic network, metabolic control. Okay, now see another difficult case. At some point of time, it is multiple input and multiple output, complete genetic network. See, it is so beautiful. So these are four diverse strategic regulations which are governed in a cell, and cell properly controls all of them. But now see the beauty. Some proteins are involved in substantial number of interactions, many different interactions, as I've shown you the big diagram. Okay, but see, it is all governed in a cell very precisely. Not even a milligram error comes up, and the moment it turns up, some disease turns up. Okay. So in this slide, we are discussing the regulatory control of three genes, how they are able to control each other's activity, and how the overall activity is governed. C here A is activating B, all right. B activates C and C again. See? But C is inhibiting A at the end. After the threshold concentration of some molecules, exceed the threshold. Okay, some the molecular threshold, molecular concentration exceeds the threshold, the reaction stops. The C starts acting as a feedback loop. So these such kind of regulatory controls are properly governed in a cell, and there are many different interaction rules to work it out. Either it's a Boolean function, that is 1:1, or sigmoidal function, or semilinear function. Like subject to different conditions, we are using different constraints, different functions to work it out. Okay. Now let us understand the Boolean networks, how the things work out. See, understand that the Kaufman, like in 1993, worked it out, the origin of waters. But he was claiming that, to understand these gene networks, we should consider the honor of states. What he meant was, within the large network, when you are analyzing a large network, some genes are tweaked on, and some genes are tweaked off suddenly. Okay, depending on certain constraints, certain biomolecular conditions. Now, when the gene is on, he should represent it as 1. And when the gene is off, he called it as 0. Only the binary states, subject to input or output. So he was calling that all this can be represented as Boolean networks. Like this. The C part is dependent on both A and B. The C is dependent on both A and B. But since C requires the presence of both A and B, only when the both are one, both are expressed, then only the C will be working out. Otherwise, the C will be 0. Simple. Okay, so there are some Boolean maps, the Boolean table. Boolean table started being utilized for analyzing the gene networks. Yes, the Boolean table of computer science background is now started being implemented for analyzing the gene networks. Now see the discussion further. If K inputs the regulatory genes, it's all depending on building the mathematical equations. Okay, so the resultant equations were worked out. Okay, so depending on the values getting changed, things will be changed, and depending on the context of different genes, the rules of different genes were changed. Okay, some genes are like 1:1, some genes are multigenic, some genes are under the control of several different cases, but all these can be represented in terms of equations. Now see here, the sum of system was the sum of values of all the nodes. Yes, so that the overall calculation can be done for the complete network, and it allows the analysis at the network level. So whenever there's a change coming up, like we constantly keep on updating the values. So the moment some molecule turns up, it initially comes, but it is then getting utilized. So the 1 and 0 states will be constantly getting changed. Alright. So it allows us to help us understand the dynamic nature of a cell. The reactions are constantly being changing the values. Okay, we can always update the values of all these enzymes. Just, just by 1 or 0 states, the complete network can be represented. Okay, by using a mathematical equation. And lastly, the equation will be interlinking all the states together by summing up the values. See, so we started building algorithms for network inference from binary data. But binary is not a good solution for binary systems, for biological systems. Can somebody understand how? I'll give you a small hint. Is the gene always on or always off? Or is the expression always even if some expression is there, or even if the expression is very high? Are the two things same or different? Think. Uh, over the 1 or 0 states of the enzymes, there's some more to it. How much the level of expression? That also defines many things for the enzyme. So this enzymatic control is very complex, and depending on different molecules, the enzymatic behavior keep on changing. All right. Okay. So only the things are very complicated. All right. Understand that whenever the things could not be worked out simply, like we started putting probability there. Okay. This part was putting up along as some sort of confidence. So relationships are beyond pairwise interactions and involve structural interactions with several other proteins as well. Things are not so simple. The overall regulation within the network involves effects of several gene products, several different proteins together. Okay, it's not so simple. So then we are putting the probability also. Under this condition, there's a probability that this thing will be on. And under certain conditions, the probability will be zero that this thing will be there. So it is not 0 or 1, but some sort of probability also. Under this condition, there's a high chance this will be there. Other times, it may not be there. This was allowing some sort of flexibility in terms of interactions. It was not necessarily linear or Boolean. Okay, it was allowing some sort of probability also that this will be allowed to interact so that the multi-genic control or when the multiple proteins are controlling a single behavior, the things can be worked out or analyzed properly. See, given a graph, see V and D, see, where each node V belongs to the V set of nodes is associated with a random variable VXV. See? And the E is the edge here. All these E's are connecting the edges. X1, X2, X3, X4, X5, X6. All these 6 nodes are connected properly. See? Now understand what is the probability that X2 will be there. It is depending on X1. Only when X1 is expressed, X2 will be turning up. Are you understanding? The X2 expression probability is dependent on the X1. So they are putting a direction like this. Understand properly. Likewise, I'm putting the thing here. X4 part is expressed only when X1 is there. If X1 is not there, neither X2 nor X4 will be coming up. Now see the case here. X3 part. X3 is dependent on X2. When the X2 turns up, which is itself governed by X1, turns up X2 turning up, then the X3 will be produced. So the X3 probability is dependent on X2. It's a called a conditional probability. Likewise, the X5 is dependent on the X4. See, the probability of X5 is dependent on X4. And see here X6. It is dependent on both x2 and x5. See, so probability of x6 is dependent on both x2 and x5. x6 cannot be expressed until x2 and x5 comes up to the required concentration in the cell. Then only the x6 can be taken up further. Alright. So now when I put it all together, see, the x1, x2, x3, x4, x5, x6 is the depending, I'm multiplying the probabilities, all these probabilities together. But this is just mathematical. Absolute biological part, I don't know how the things will work out. Or whatever the way I'm applying the probability here on x6, is it same as x2? Or put liquid weights on all of them, is it right way in a certain cell or not? I don't know. Because depending on different cells, different things, the biological part may change. But still, yes, the probabilistic graphical model are analyzing this complete this biological reactions this way. Okay, so as to get some insights out of it. All right. So see, so only like the mutual information part has been taken up further, MI, and it has been long been worked out. Different servers are turned up for MI prediction, mutual information prediction. All this is working as what is the part that XY turns up together. If the probability of like probability of expression of X and Y is like this, X express, Y express, and XY turning up all together. They both turn up. What is the reaction further? So for the reaction further, so the subsequent reaction to occur, the Px and Py should be there over the required constant. Okay. So if I say then the probability of X, probability of Y crosses 0.7 and zero point 0.9. Okay. Then in this case the things works out. It may be there. Okay, but I'm just assigning a confidence interval. So the log aspect is there, see, and the Px and Py, see. What is the probability of X and Y? So it depends on all the X, all the Y's. How the things can be worked out. A high value is a high predictability, and a low value is a low predictability. Means both the things do not work together. They are completely independent. Okay, I hope I am making sense. If both the two genes X and Y do not follow each over each other, like for getting a new function, if both X and Y are not needed to decide a new part, new expression part, new molecule production, then I can say like they are completely independent. All right. So we are analyzing several different parts like this, like computing statistical queries regarding the network. What is the probability of X being true if Y is false, Z is true? Under different conditions, the fate will be changed for certain molecules. What is the most likely assignment of all the values of the nodes of the network? I can do it. Okay. Like general purpose. Now see. So when I am building a computational cost, depends on the topology of the network. Okay. More edges are there, more like different molecules are combined together, and my computational model for all the enzymes together includes multiple enzymes, multiple edges for a big graph. It will take me a lot of time because there's a multiple numerous probabilities coming up, like the conditional probabilities. Numerous probabilities will be coming up. If this happens, what is the next part? If this happens, what is the next part? And the system keep on computing all of that. Okay, so now I hope you understand this is so much complicated. If you have understood so far, let us answer these MCQ questions. In a Boolean probability model of a metabolic network, the Boolean variables represent. See, the Boolean variables represent the concentration level of different metabolites over which the probability is applied. The probability that it is actually coming up to that aspect that is level or not to guide the required function. Okay, so Here, stoichiometric coefficients of reactants and products, this is wrong. The likelihood of metabolic reactions is wrong. The concentration levels of metabolites, yes, this is the right answer. The kinetic parameters of enzymes, this is wrong. So the Boolean variables, it means the concentration levels of metabolites. Alright, now see the next question. In a Boolean probability model of a metabolic network, the states of a Boolean variable represent. Now the states means whether it's coming up or not. The concentration is there, whether it's actually coming up to that part or not. Means, is it being activated or not? Remember 0 and 1, the binary part. So, the concentration levels of metabolites, this is not the right answer. This is wrong. Stoichiometric coefficients of reactants and products, again this is wrong. The flux rates of metabolic reactions is also wrong. The activation status of metabolic reactions or components, yes, this is the right answer. So when we are telling about the status of Boolean variables, the status, okay, the status means activation status. Whenever it's coming up, if it is coming up, the Boolean variable overall represent the concentration part. Coming up or not coming up is the activation status. The status of being activated to guide the function the same way. Over which the probability is there. Alright. I hope you might have understood properly now. Now let us answer this conceptual question. In a Boolean probability model of a metabolic network, what do the probabilities assigned to the Boolean variables represent? The probability means whether it's actually coming up to that concentration level or not. Okay, concentration defines the Boolean variable. But if it's actually coming up to that level, okay, it's initially 0. But if it's actually coming up to the required concentration or not, the probability tell it. For which the probability is calculated. You are calculating the probability only to check whether it actually may come up to that level or not to guide the next function. Okay, so only we are multiplying all the probabilities at the end. The first step came up to the required probability, required expression level to guide the next function. And same goes on for the later enzymes. Every enzyme is producing the required impact. Every enzyme is coming up to the required concentration level for all the metabolites. Then only the things can work out. Alright. Now, in the last part of this session, let us understand how to analyze these complete networks. Okay, or how do you actually represent them? Okay, so that even if for many enzymes, many molecules are present, you don't get confused. Okay, so you can follow up some set of rules, and through those rules, you can happily analyze all the enzymes, all the molecules together on a big network. Okay, so see this part. How do you actually represent all the metabolic network? Okay, the compounds are shown as boxes here. See, different compounds: polymers, glutamate, octoglutamate, pyruvate, alanine, D-alanine, L-alpha-alanine, L-aspartate. All these amino acids in this. See, I taken it from Mikal's 1993 book. See, the poster which was presented. See, the arrows connect the compounds to show interconvergence. The arrows coming up with the directional pair. I'm putting up a direction also here. Which molecule is producing what other molecule? Understand? Which molecule is producing what other molecule? I'm not simply connecting them just to show their connection. That this enzyme is somehow linked to the other one. No. I'm also showing which molecule is properly producing the other one. Okay. And these arrows are then labeled with the name of particular enzyme also, which is which is associated with that catalysis of that particular substrate to the product. Some enzyme is constantly producing that required product. Okay, so what is that enzyme? See, aspartate decarboxylase. Okay, see, all these parts. Alanine decimase. That this is a reversible reaction. See, okay, the asparaginase, bromellar asparagine to the aspartate. See, but it's properly being converted, and I'm showing the enzyme also. So the all the molecules are shown in the boxes. I'm encapsulating them with the boxes. Then I'm putting up the direction with the arrow that this side to this side the reaction is taking place, and then I'm putting the enzyme also on the top of the arrow that this enzyme is catalyzing or mediating this reaction step. And if there are cofactors needed, I'm also putting the cofactors in the curved arrows. Okay, see here, different curved arrows. See. Uh, See, glyoxylate is produced here. See, like glyoxylate is required here. See, NADP plus is required here. H2O is just the water part. And see, COSH, NADP plus, all these I'm showing with the curved arrow. See, Na2, ATP, all these parts. Everything can be shown in the curved arrows. If it is coming in, like within the reaction step, it is just used as an input. If it is coming outside, the direction is towards outside. Okay, like this. If it is coming inside, this is required in the reaction step. Okay, the reactant to the product catalyzed by the enzyme. Okay, so if something comes in, I'm showing the cofactor here. If something comes in, it is needed. If something goes out, this is produced. Like that, I show the complete metabolic network. Okay. So, how do you actually build up the metabolic network? How do you, what all molecules or what all the information that is that you should consider? So, remember that whenever we are building up all this, we need the genome annotation for all the enzymes coming up. What is their functional behavior? Okay, biochemistry of reaction. That under what condition, what thing they are doing the reaction. Okay, so only we can get to know about the cofactors involved. We should know the biochemistry, physiological state of cell. This allows us to analyze different molecules, different conditions which the cell is undergoing. Okay, so that if something is missed out in these conditions, it can be considered here. Okay, and then for reactions, whatever the things you have got so far, you have all the reaction data sets, everything, all the reactions with you. That all these reactions are going on for the considered set of genes. All right. So now you understand that what are the reactions there. If something is missed out, you put it also. All right. So then you build the network. You connect them all together, and then we build the metabolic network. We completely build the metabolic network. Metabolic network is built here. So the network construction is just a preliminary step. But then we finalize the metabolic network using the analytical methods by proofreading different steps. Did I see whatever I am connecting, is it actually there or not? Okay, so see here for different species, number of genes, number of metabolites, number of reactions, and when they are published. I've just taken it from a research article. See, so so many reaction steps after so many reactions, this complete network of 708 genes, okay, could be connected. All right, it's so much beautiful. Whenever I'm building a graph like this, can I make it simple graph like this, metabolite? You know we have been discussing the directionality within the network, which molecule is able to produce what other molecule. So I'm not putting the things here. So means this graph is not correct. digraph where I'm putting the network also, the directionality. But is it right? Is it right? Think. Not at all showing which molecule is what. Okay, is it a reactant? Is it product? Is it changing the conditions in all those parts? See, I'm putting the bipartite here. Okay, because it is putting up different cofactors also. This is producing this through this cofactor. I'm putting up the things. But you understand that all these enzymes do not behave the same way with different reactants, different enzymes. The moment two enzymes comes closer, their complex may change the behavior completely as per the need of the cell. So it is not simple graph. Okay, it is not digraph. It is not bipartite graph as well. Okay. It is much more complicated, where different molecules turns up to change the complete behavior. Okay. So within the complete network, if I am saying how do the kinetic model work it out? Okay, that is the time-dependent reactions. Which molecule is able to produce which other molecule and how much is being produced per unit time. Okay, the complete production rate. Then we are using all the for the kinetic models. We are working on the metabolic concentrations, the enzyme concentrations, enzyme activity rate, everything. Under what phase of the cell the complete exponential phase is going on. Okay, depending on all of it, we work out a complete set of differential equations. Okay, for different substrates, different conditions, different enzymes. Then after that is done, okay, it requires a detailed data about the several enzymes. As we have been discussing, what point of time the enzyme is being produced, like how much time the enzyme is active there, all the details are required. And once we have everything, we are able to properly map how the kinetic model works it out. Okay, through which the flux balance also can be properly worked out. How much concentration of the substrate is being passed on further in each step to give the required product at the end. Okay, and what is the production rate of each enzyme? We can exactly work it out, and this allows us to find out the rate limiting enzyme as well. All right. As I've been showing here, that we are interested in working out the topological answers to put the complete reactions of all the enzymes products together at one paper, one shot. Okay, so I'm putting the compounds, okay, unique substrates, the unique products, all of them together. Nothing is duplicated in the paper. I'm putting up all throughout and then connecting them with different arrows. The categorized reactions and putting the enzymes over the top of the complete reaction, the arrow which is there. Okay, so remember, we can put it up this way throughout all this complete set of information. We can put it up. Even if a big complex diagram is there, we can put it up on a single paper and then analyze the complete data. Today I'm showing this diagram here on the left. Okay. And it's so many equations are being worked it out here. I just taken it randomly from like complete biological metabolic network. This is working out. See, if I am calling it elementary flux mode here, this is just the basic definition. How the things work steadily, independently at a steady state. Okay, how they can operate. This is just for different one enzyme. For each of the enzyme, it can be elementary flux mode. Okay. Or even if I'm connecting two at a time, however the steady state they'll operate. But once I'm telling flux balance analysis, then I'm only telling that I'm predicting the reaction rate, predicting the reaction flux. How many molecules or products are formed from the reactants? Be it the set of one enzyme two enzymes or multiple enzymes, or all of these enzymes together at a time. Okay, that's the part. But all these parts worked out to kinetic parameters. So again, so that we can effectively work out the flux part. Okay, so even if some molecule is changed a bit, my flux balance analysis, the FPA part is very sensitive enough to detect the yes, this molecule is changed. The concentration of this molecule is decreased or increased in this much time, because of which the product the product formation rate for certain molecule will be drastically changed, either it will be increased or decreased. Okay, but it is very sensitive. So that's the entire beauty of all the reactions. And once I'm able to connect all these parts properly, I've done my homework properly for the complete part, then I'm able to effectively match each and every part. The moment I'm able to take out some molecule, my FB analysis will exactly tell me that yes, this concentration has gone down, because of which the product formation rate of some molecules will be either increased or decreased. Okay. See another example of flux balance analysis. Okay. I've taken it from this research paper. This research paper taken from. The researcher shows that when there is no oxygen, complete no oxygen, anoxic conditions, see how many enzymes are produced and how they are working it out in cytosol and plastid. But the moment when the concentration is hypoxic, the conditions are hypoxic in the cell, that is limited oxygen. Then see, are you seeing here some sort of part, some changes here? See, the some things which are produced too much and now the concentration is decreased. This was not at all there in mitochondria. Now this also started. Why? Because oxygen acts as a terminal electron receptor. Okay, the TCA cycle also started. Earlier only this was cytosol and plastid, no mitochondrion. Only the glycolysis was going on here, if you can see clearly. I hope you might have observed. But now, when the oxygen comes up here, the TCA cycle started properly. See, it worked it out, but not at the very efficient rate, because oxygen is less. But still, it started, and the channelizing enzymes also started working it out. But now, when the oxygen demand is not very high, the oxygen demand is too much, and we are getting unlimited oxygen. See? All this TCS cycle is completely running at full flow. Okay. See? And all these intermittent paths, intermittent enzymes are coming up at their complete bloom. Okay, they are effectively expressed. So it leads to the complete change in the behavior of several enzymes just by the presence of oxygen at an unlimited concentration. So this is the entire beauty as such. Okay. So the moment something is changed a bit, many things are drastically changed in the cell, and all this can be predicted or analyzed by the FBA part, flux balance analysis. I hope you might have now understood. Now, understand that we have been discussing about different rates, but these different rates are not static, they are dynamic in nature. All the times, different substrates or different molecules are always getting produced, so their concentrations are always getting changed within a cell. So, if suppose this ds1 by dt, that is the production of S1 molecule within a cell, is V1 minus V2, means V1 is the reaction rate at which it is produced, and the reaction rate at which it is getting utilized is V2. As in this case, see, from initial substrate X0, it is producing S1, then S1 is produced at S2, then S2 is being utilized to produce the last molecule X1, final end product. X0 to S1 to S2 and then X1. And here the reaction rates are like V1, V2, V3. DS1, what is the concentration level of S1? What is the change in concentration of S1 per unit time? Is V1 minus V2. V1 is the rate at which it is synthesized and V2 it is being utilized. V1 minus V2. So suppose the S1 is produced at 5 mg per liter, but 2 mg per liter is being utilized later on, immediately after it is produced. Means the production rate is only 3 mg per liter. 5 mg per liter minus 2 mg per liter. Likewise, for S2, ds2 by dt is like V2 is being produced, V2 yet. So V2 minus V3. And V3 is the only rate at which is X1 is getting produced. So X1 production rate is only V3. But all these are not static, they are dynamic in nature. Okay, all the molecular functions, molecular networks, the regulatory controls, the protein-protein interactions are all dynamic. Okay, like the substrate concentration changes, product concentration change, enzyme activity changes, everything. Okay, subject to different parts. Only the systems biology markup language have been developed, which allows us to optimize various parameters. Okay, like the equations of all this, I normally put in deterministic or stochastic. Like that, I will consider this as deterministic means something where you can easily find out, easily quantify based on the given conditions that this is going to be like this, like Newton's laws. Okay, but stochastic means it is just a prediction based on these constants. This thing could be there. Okay, but this thing will be there, not possible. Deterministic means subject to these conditions, this thing will be there like this. Deterministic, like Newton's laws, fixed. Stochastic means under some conditions the things may work out like this. Alright. So only understand that we have been developing many different algorithms so far. So for modeling the network function, many things have been worked out. I'm putting it like this. Whenever it is kinetic model or approximate kinetics, okay, where the accuracy is highest, because it is being derived from the mathematical part. All these experimental data also worked out, everything done. So it requires kinetic constants, which are mostly unknown. Okay, which are mostly unknown. Just like if I'm considering the lesser number of enzymes, I can always get it very easily. I can still do it. Okay, I can still work out. Okay, when the enzymes are lesser in number, okay, small number. Enzymes are smaller in number, still work it out. So there the scale is lowest. Lesser number of enzymes, accuracy is highest. So everything will be properly worked out. Okay, but the moment we have the very high number of enzymes. Okay, I'm using the graph theory. Okay, with the structural network properties, degree distribution, centrality, like how many times the different points in the graph is properly connected, how many times some node in the graph, enzyme in the graph is connected at the center, at the key regulatory hub. Okay, when the like, many number of enzymes are present, the topological analysis comes up. When the scale is there, see, very high number of enzymes. And under these cases, okay, when large number of enzymes are used, the accuracy is lowest. The accuracy is lowest accuracy. Here this is very highly accurate. So this defines the highly accurate part when small number of enzymes are present. But this defines like very less accurate. Okay, robustly they are there, we can still get it some part, but they are still not that much accurate. Okay, but here the large number of enzymes are used. So in between these, the probabilistic model comes up. conventional functional models, okay, probabilistic models, okay, where I'm knowing able to predict some things for large neural networks. Okay, they are constraint-based models. Now these are having some a bit higher number of enzymes, not lowest, not few, just few. I'm still having some more enzymes. So there the optimization theory comes up, like constraint space of possible steady state behaviors, okay, constraint network behaviors, everything. I'm trying to consider every single parameter here. So kinetic models, approximate kinetics, constraint-based models, conventional functional models, and topological analysis. This is a strategy which I'm completely working out to make the part properly. Going from the smaller number of enzymes to a large number of enzymes. So now if I ask you what is the modeling strategy for constructing and designing the biochemical reaction network, understand that there are the databases there. Okay, different databases are used. This is the prime source for all the information. Select the genome atlas, the complete reaction part, mine database, all these parts come up here. Once they are there, I'm able to build the network. Different categories of network. See, substrate, bipartite, hypergraph, reaction graph, and all of them. Even the reaction matrix also. See, the binary network. I hope you can understand now. See, but understand that all the nodes in the graph are not that much important for me. So whenever I'm doing something, I discard all the outlier genes, outlier genes which are not making sense for my small pathway. I'm not able to like analyze the complete big network for the single part. Okay, so all the interaction partners for an enzyme which are not making sense for me, I discard them. This is termed as network pruning. Here I am considering I checked out the sequence homology for my protein, and then through different species I work out their networks and then understand the implied network in my species. Okay, when the Tanimoto coefficient also helps me. The structural similarity of different ligands. All the network part will be there. So all the outlier part, all the outlier connections which are not making sense for me, I discard them. Then once it is done, I work out search algorithm. Okay, that what could be the required reaction network for under this condition. I apply the depth first search, breadth first search, k shortest path. All these are parameters. Like all these are like statistical scaling parameters to connect various enzymes in a part. Because all these enzymes are just like nodes, and the edges are able to connect them. So the part is objective is to connect them the best possible way. But I hope you understand that when I'm building up, connecting up different parts, different all these parts, so I'll be getting multiple solutions. I'll be getting a multiple solutions with similar high scores. I rank the pathways, I rank the graphs, which is making sense for me. Okay, so to get the required path, so based on the number of reaction steps, thermodynamics, product yield, like metabolic toxicity, through all these parameters, I'm able to rank the part, rank the reaction network. Okay, this is along with the work it out. Strategies, network information is normally inaccessible. Reaction path is not known. So known enzymes and reaction steps are first mapped. And then documented for the balanced equation form is first chemically and physically analyzed in terms of the number of molecules, required time, and surface area and volume of the active site. And all of these parts are considered. I get different databases, I try to build the network, I try to discard the outlier part, outlier enzymes, and then for all of this, I try to search the best possible reaction network to maximally connect all the leftover enzymes. Okay, then once it is done, and if I'm getting multiple solutions here, I try to take the best solution. I discard all the other outlier solutions. Okay, this is the normal strategy which is worked out. If I'm putting the same part in this way, my task is given the genome information for an organism, I need to find the metabolic network. For this, I can use sequence databases, genome annotations, database of metabolic reactions. So, using all this, I need to figure out sequence, like sequence similarity search, text extraction, machine learning, experimental data. Through all this, I should be able to build the required network. Okay, that is the only part. So whatever we were just discussing in last slide, I'm now showing the exact part as a question here. Depending on what is the input, what is the required output what I want. Okay. So see, metabolic construction from a genome annotation is worked out like this. See, this just as an example. Glycolysis part, different parts. If you take up the cake part, you'll get the things like this. If you work it, zoom it out here, different enzymes are shown here with the green color with the enzyme EC number, enzyme commission number. So that you can work it out, read more about them later. Between EPSP synthase and shikimate kinase, different sequences are still conserved. I can get the sequence patterns. Non-ligand binding from bacterial ATP GTP binding motifs. Once I'm, Once I'm aligning the known data, I can get the information. All these parts is used to interpret the homology in different species, different sequences, so that their reaction network, their reaction kinetics can also be exactly like analyzed in the other part. Whatever the reaction network is working here, exactly the same reaction mapping can be worked out for the other sequence also. Okay, because the motifs are exactly matching. We know that this enzyme is also going to be EPSP synthase or shikimate kinase. All right, but now we know that it's like the prediction part, like all the prediction algorithms have got some limitations. Same goes here. Okay, there are large evolutionary distances are there. So every time when I'm transferring the function from some sequence to the other sequence, like specifically for the mapping of the reaction network, it's because if the distance becomes too large, if the two sequences considered are very distantly related in evolution, whatever the reaction network, metabolic network of one sequence cannot be transferred on the other one. I cannot consider that way because they are very diverged very earlier in the evolution. Okay, they are evolutionary too distant, so their reaction network cannot be analyzed same way. Their reaction network cannot be the same or cannot be too similar. Enzymes may be too divergent from recognized from the sequence. Okay. The enzymes may have got multiple functions. Some enzymes multiple protein domains that have different functions. So things may be really complicated. Okay. Whenever one enzyme is able to work out multiple activities, we call it that enzyme like is a moonlighting enzyme. That is, it is able to catalyze multiple different reactions using the same active site. Alright. Reactions with unknown sequences. This is another big trouble. See, many times we don't know many things about different new enzymes. So for all of them, we are unable to build their network properly because we don't know the enzyme activity. unknown reactions, see? And the reactions are itself unknown. Okay. So we don't know how their metabolic network will be there. Okay. Because we have not characterized these sequences so far. So what is the take home message from the entire session? See, read it carefully here. In a Boolean probability model, metabolic network is represented using Boolean variables that can be either on or off, representing the active or inactive state of reactions or components. This is an important part, on or off. The model assigns probability to these states, capturing the likelihood of their activation. Understand the words. I'm only capturing the likelihood of their activation using probability based on experimental data or prior knowledge. Okay. The local relationships or the rules describe how the states of variables influence each other, reflecting the network's connectivity and regulatory interactions. By simulating the model, predict one can predict, okay, variable states under different conditions to identify critical pathways. Okay, we can also find out gain the qualitative insights on the system's behavior. The complete system's behavior, we can get the qualitative insights that how it is working like this. What could be the reasons? Okay, Boolean probability models offer a very simplistic yet informative representation of metabolic network analysis and understanding. For a very simplistic understanding, Boolean network is very important. Well, a very good as a very initial start, we can analyze the network that way to build up initial understanding. Okay, all right, and thank you so much for your patience. If you have some questions, you can always mail me. Goodbye.